Thank you. And this is a little different, as you'll see. So I've been a professor for, make sure this is as high as it can go. I've been a professor for 42 years, which means a few things. First of all, although I still find excitement in the ways data analysis can advance science, I'm getting pretty old. In fact, I was recently diagnosed with CRS, can't remember shit, which is one of the reasons I now script my talks. Well, uh-oh, why did that happen? While I've adjusted very comfortably to almost always being the oldest person in the room, it has its disadvantages. As a teenager, I was already an absent-minded professor, and at my advanced age, it's become downright dangerous. A few years ago, I was at an off-campus meeting and walking back to my car deep in thought, and I got in and I had a moment of panic because the steering wheel was gone. And I thought, who would steal a steering wheel? And then I realized I'd gotten in the back seat. So I wish I were kidding. Aside from the script, the consequences of age are, for me, not very great. As a workshop lecturer, I've always thought about the ways research results can illustrate general lessons for reasoning from data. And I've spent a lot of time trying to articulate and share those connections. The only thing I've noticed recently is that the big picture now seems even more important. So with that in mind, I'm going to discuss statistical assessment of interaction across brain regions from multi-electrode recordings. That's what's different, by describing the problem and my interest in it, and then stepping through 3 examples and identifying in those individual pieces of work some of the more general statistical teachings and strategies that were brought to bear. And then finally, I'm going to say a few words about my continuing interest in these high-level concepts. So as you know, the brain sciences seek to understand how neural activity produces behavior. It's a fascinating subject because our brains define us, and they may be considered to operate as information processors, which opens the door to many kinds of technical analysis. Historically, and through to the present day, the biggest influences on theoretical neuroscience have come from information theory and statistical physics, especially through differential equation style modeling, with computational neuroscience, which now includes advanced data analysis, being dominated by those trained in physics, while only a small handful exhibit what we would call statistical thinking. This was the landscape I found myself dropped into 25 years ago. And it's remarkable how very few statisticians or others who think like statisticians work in neurophysiology. At the same time, anyone who ventures into the field, as I did, is struck both by the inspiring progress that's occurred, especially over the past 70 years, and by the brain's daunting complexity. In fact, as you follow the emerging literature, it seems that every promising discovery inevitably reveals new, previously unrecognized subtleties. I was keenly aware of this reality when, a few years ago, I wrote my most recent and final renewal proposal for a grant that's funded by the National Institute of Mental Health. It's been funded since 2001. In response, I wanted to articulate an ultimate goal of such basic research that, even if it's long-term, seemed realistic. And I latched onto a simple operational suggestion. I don't like that. Can I okay this? Let me just try that. All right. The operational suggestion was, so what I pitched was the motivation for the research. I pitched by saying that new statistical methods could lead to improved treatment strategies involving interventions that would modify brain circuits with precision in space and time. It seems to me that 50 years from now, treatments for mental health disorders will have advanced far beyond where they are today. But to get there, neuroscience will have to go beyond this is not working now. Should I use the keyboard? I can use the return. Do you think it's going to start working again? Okay. To get there, where was I? we'll have to go beyond these simple static circuit diagrams that depict interactions among brain areas without describing the flow of information while an organism accomplishes some behavior. Improved characterizations would use timing relationships, consider both local and cross-area computation, and describe cross-population dynamics. So I've been interested in all of these things. The data we analyzed in what I'm going to show you today were collected from many types of multi-electrode arrays, 3 of which are pictured on the right. As shown on the extreme left, each electrode records voltage signals from many nearby neurons. Cutting-edge data analytic methods are being developed by many people, especially in conjunction with the so-called Utah array and the neural pixels array. The latter, especially, has sparked interest because it packs a lot of electrodes densely into a small area. A typical neural pixels array has more than 1,000 electrodes available for recording. Several of our most recent papers, and several still in progress, have been based on data collected at the Allen Institute by Josh Siegel, who's managed to record from 6 neural pixels simultaneously, each with more than 300 active electrodes in the brains of mice, while they are shown a variety of visual images. Speaking as someone who's been working on analysis of neural recordings for roughly 25 years, it's an exciting time. We're finally getting routinely the kind of data that we started to envision more than 15 years ago. And for the past 8 years, all the trainees working with me have concentrated on identifying cross-population interactions, typically, though not always, from different parts of the brain and their timing. Specifically, we've examined spike trains and local field potentials, as I'll explain. All the papers I mentioned today can be found on my website, and I will also briefly refer to that book that I published in 2014 with 2 co-authors who helped in smaller but very important ways. Emery Brown, who's both a PhD statistician and an MD specializing in anesthesiology, with endowed chairs at both MIT and Harvard Medical School, and his former PhD student, Uri Eden, who's now a full professor at Boston University. Speaking as a statistician, many analyses in the literature fail to capture potentially important information in the data, and in the opening section of the book, we said, we have been struck repeatedly by the absence of what we might call statistical thinking, or application of the statistical paradigm, and a resulting loss of opportunity to make full and effective use of the data. Following the research that a series of trainees and I conducted in the past 8 years, along with a foundation we helped lay across the previous 15 years, last year we published a review on identification of interacting neural populations, which had 176 references, and 25 of those were to our own work. We sandwiched our list of methods between an elementary overview of the statistical paradigm and a list of high-level issues. In the introduction, we said our discussion aims to guide thinking about the linked criteria of physiological relevance and statistical standards of evidence. And at the end of the paper, we emphasize the importance of connecting the statistical methods with the physiology and the inherent challenges of doing so by saying scientific conclusions must combine pre-existing knowledge and assumptions with data analyses based increasingly on advanced methods that involve subtleties of reasoning. Here are the 3 studies I said I'd talk about, all based on spike trains or local field potentials, recorded from multi-electrode arrays. So let me get everyone on the same page by reviewing the basics of spike trains and LFPs. Neurons communicate primarily through rapid electrical discharges known as action potentials, or spikes, that's what we usually call them, which take place over approximately 1 millisecond. This figure is a voltage trace recorded across the cell membrane, though all of the voltage recordings in the data I'm talking about are recorded by an electrode outside the local neurons, as illustrated in this image, which I showed earlier. This figure was created by former Carnegie Mellon student Ryan Kelly, who became my postdoc for 18 months before going to Google. Panel C shows the raw voltage recordings from all the electrodes on the left, along with a pair of displays on the right, based on a trace from one of the electrodes. The top part of panel C shows a low-pass filtered version of the voltage. In statistics, we call it smooth, which, by the way, I assume is a term we got from John Tukey. And it becomes the local field potential representing the aggregated neural activity from hundreds of neural synapses in the neighborhood of the electrode. The lower part of panel C shows a series of action potentials, or spikes, overlaid on top of each other. This results from a clustering procedure after high-pass filtering. One set of similar waveform curves is in red, another is in blue, representing spikes from 2 neurons recorded on this electrode. Below them are the sequences of time points corresponding to these waveforms, which constitute the 2 neural spike trains. Here on the right, I'm showing you the variation of neural firing patterns, the spike trains, across 3 repetitions of the experiment, called trials, and across time and across 4 neighboring neurons. It comes from an earlier review I organized, a large international effort aimed at summarizing concisely both mathematical and statistical modeling approaches. By the way, that got started at the SAMSI meeting that you referenced. That's the origin of that very substantial review paper with 25 authors. Neurophysiologists do their very best to make sure the experimental trial replications are as similar to each other as possible and independent. Like the bulk of the literature until very recently, we have focused our attention on these repeated trial experiments. The main interest here is in the frequency with which the neurons spike. In other words, the neuron firing rates. For example, particular neurons fire rapidly in response to specific visual stimuli. There are simple ways to reduce the variability. One can average across trials to produce a peristimulus time histogram, or PSTH, which will come up again later. Or we can average across time, which creates spike counts, and that's the most common summary analyzed in the neurophysiology literature that's relevant to what I'm talking about. I want to mention now some results on spike count correlation, often called noise correlation, which refers to the correlation across repeated trials from among the spike counts from pairs of neurons. These histograms display the variation of correlation measures across substantial numbers of pairs of neurons. You'll notice that while there's a lot of variation, there is on average a small positive correlation. This is a commonly observed phenomenon, and its relevance to the functioning of circuits in the brain has been widely discussed, beginning especially with the 1998 paper by Mike Shadlin and Bill Newsom, who used additional data to pick the Pearson correlation value of 0.12 as a typical value in their very influential article, which I use as one of the first readings in a course on computational neuroscience that I developed and have taught almost every year since 2011, called Statistical Models of the Brain. These guys are both famous, and at this point I know Mike Shadlin pretty well. However, the peristimulus time histogram and the spike count correlation histogram lose information that could be useful. The first about trial-to-trial variation, and the second about precise spike timing. And another point, the consequence of which I'll note in a moment, is that spike counts are subject to Poisson-like noise. The usual approach any of us would take is to formulate a statistical model and use it to consider what we want to learn from the data. By the way, I could emphasize this point by saying that as much as I am immensely proud of some of our papers, I don't consider any of the stuff we've done to have been especially clever. I've just followed what I learned statisticians are supposed to do. Statistically, spike trains are modeled as point processes, meaning events that occur randomly across time, and these produce Poisson-like noise in the sense that they're not quite Poisson processes, but they are, at least in the cerebral cortex of behaving animals and often in other parts of the brain, pretty similar. Okay, finally, we get to the first study I want to talk about, done in collaboration with Josh Siegel at the Allen Institute, as I said. I already showed you this figure illustrating the placement of 6 neuropixels in the mouse visual system. The lead author was my PhD student, Yu Chen, who graduated in 2021. He analyzed the spike trains from dozens of neurons recorded from each of 3 areas in response to drifting gratings, as pictured in the figure. You can see that mouse looking at these drifting gratings, with 15 repetitions in each of many experimental conditions that define the orientation and the frequency of the gratings. Drifting gratings are well known to drive responses in visual cortex. Here, I'm going to show you a few results involving just 2 areas, V1 and LM. V1, also called primary visual cortex, and it feeds into higher visual areas such as the lateromedial area, abbreviated LM. Panel A shows what amounts to 2 smoothed population-based peristimulus time histograms fitted with non-parametric regression, which are firing rate curves in each area, effectively averaging across all the neurons in each area and across all the trials. The y-axis says spikes per second, but it's understood in this context to be spikes per second per neuron. We called the two dominant peaks population bursts and labeled them peak 1 and peak 2. The first peak, shortly after stimulus onset, presumably is dominated by feed-forward connections from the eye, to the ganglion cells in the thalamus and then back up to the visual cortex. But the second peak, which is much later, presumably involves both feed-forward and feedback connections from other brain areas. I'm going to focus on the timing of peak 2, specifically the time at which the peak 2 maximal firing rate occurs. I was interested in peak timing because when many neurons in a population start to fire rapidly, we can presume that information is transmitted. And from a statistical perspective, by definition, lots of spikes occur near the time of maximal firing rate. So the time itself should be relatively well determined statistically. In particular, I was hoping that with some effort, we could find much higher correlations than those small ones I showed you earlier for spike counts, by instead examining the correlation of peak timing across the two areas. And that's what actually happened. The project really got started with a pair of undergraduates, Brian Medina, who's now a PhD student at MIT, and Hannah Douglas, now a PhD student at Princeton. I asked Brian to create population PSTHs for each trial and smooth them. So that's similar to what's shown in panel A, except that the data for the plots in panel A use all the trials, while what Brian did was create an a bunch of analogous smooth histograms for each trial separately, and then find the time of the peak 2 maximum in both V1 and LM, and then plot the time of the maxima for V1 against the time of the maximum for LM. So the results are shown in panel B. Again, each dot represents a pair of population peak 2 times on a particular trial. To my dismay, there was no visible association, and it turned out no statistically significant correlation, even with a pretty substantial number of trials. We had 600 trials in total. So I figured this was a job for a PhD student. Meanwhile, I asked Hannah to look at the responses of individual neurons in each area, and she documented multiple distinct kinds of responses. For example, some neurons went quiet for a short period after the stimulus onset. So the upshot was that neural responses in that area are actually quite inhomogeneous. So Yu Chen did a lot of work on this and was able to produce the plot shown in panel C, which we labeled denoised as opposed to the naive panel in plot in panel B. So there's now a remarkably high correlation, especially for anyone used to seeing those relatively small spike count correlations of the type I showed earlier. He estimated the correlation to be 0.87 with a standard error of 0.025. The other striking thing about this relative relationship is that the time lag from the peak in V1 to the peak in LM is very consistent across trials at 9.6 milliseconds with a standard deviation across trials of only 2 milliseconds, even though the trial-to-trial variability in peak times themselves in V1 and LM is large, with standard deviations of about 30 milliseconds. So you can see this large variability across trials in the plot. If you just, there's this large range of values. If you just, in your mind, project the points down onto the x-axis for LM and the y-axis for V1, it's a big range. So there's lots of discussion in the literature, often using theories about dimensionality reduction and sources for these theories. There are theories about the sources of spike count correlation. The interesting thing here is that in Yu Chen's model, the only sources of trial-to-trial variation are at the population level, with the only trial-to-trial varying parameters being the peak 1 time, the peak 2 time, and the average firing rate. Yet, after a referee asked us to look at spike count correlation histograms for individual pairs, which we did within each of the 3 areas and across all the pairs, the model fit, shown in black here, agreed quite closely with the spike count histograms. In other words, population level variation in timing and overall firing rate can produce the kind of spike count correlation we see in data, even if the neurons are otherwise firing independently of each other. The model itself has a general form that's familiar to anyone who's ever worked with Bayesian hierarchical models. It includes multiple familiar components: model-based clustering, non-parametric curve fitting, latent variables, time warping, endpoint processes. This model would be complicated for a statistical novice, but these days it's pretty straightforward for a statistics PhD student. It just takes some diligent work for a few months. Conceptually, it's straightforward. Each population is condition specific, so the orientation of the drifting, as the orientation of the drifting gratings changes, the neurons chosen to define the population changes, and the population has a condition-specific template for the way the firing rate evolves over time. Both a composition of the population and time-varying firing rate template are learned from the data. Actually, the population PSTHs for particular experimental conditions tend to have sharper peaks than the original PSTHs that I showed earlier, which were based on merged data, because the merging of conditions blurs the firing rate response functions. In the example shown here, peak 2 is very sharply defined. It's actually sharper and taller than peak 1. Also, if you look at the y-axes, the firing rates per neuron are much higher in the figure below, because the model selected the relevant neurons that were responding to predict particular experimental conditions. So the take-home messages were: first, this approach is based on relatively homogeneous populations. Second, the 3 trial varying parameters, peak 1 time, peak 2 time, and the average firing rate, are all features with high data content and thus estimated well. And third, there's a population level point process model that is a point process model for the spike times effectively merged across this relatively homogeneous set of neurons in each population. So point processes are statistically very natural models for spike trains. They're flexible, time-varying spike count models where the spike count time bins are short, 1 millisecond, so the counts themselves are either 0 or 1. From a scientific perspective, point process models allow us to get millisecond precision. And in addition, the use of a point process model will automatically correct for attenuation of correlation. So if you're not familiar with the attenuation of correlation terminology in statistics, it's about 100 years old. And it's explained by this figure, where we first generated a batch of highly correlated data shown as green dots, and then just added noise independently to each of the 2 coordinates to get the red dots. The 2 gray lines are indicating where, points that correspond to each other when the noise is added. This is very typical of spike train data due to their large Poisson-like noise. But it's easy to correct for it or denoise the correlation, as we had actually showed in a paper from 15 years ago. And again, this kind of point process model will do it automatically. In a paper we're now revising for the Journal of Neuroscience, a current graduate student, Talani Olarinre, created a much simpler approach. Yu Chen had built this complicated Bayesian hierarchical model. Talani instead decomposed the model into 3 separate steps that retain the key features of Yu Chen's approach and does almost as well, while being much easier for other groups with less expertise to use. And because it's all like not one big model, it's 3 separate pieces, it runs about 10 times faster, which was good for Talani's work because he could then analyze data from a whole bunch of mice, not just, I showed you data from 1 mouse so far. The second example I'm going to discuss involves the problem of identifying coordinated activity of LFP oscillations, which on which we collaborated with the well-known MIT neurophysiologist Earl Miller and one of his postdocs. It's based on some excellent work by 2 former PhD students, Natalie Klein and Josue Oriana, that produced a body of theory and methods we call torus graphs, for reasons that will soon become obvious. Here I'm showing you an illustration of LFP oscillations and some other data where the raw LFP is smooth, which is to say filtered within a narrow frequency band. And just in case you're unaware, it's very easy to create neural circuits that oscillate using excitatory and inhibitory neurons for intuitive reasons, starting with just 2 neurons, 1 of each type, that are connected to each other in both directions. If we drive the excitatory neuron, it starts to fire, which then drives the inhibitory neuron, and that stops the firing of the excitatory neuron. But when the excitatory neuron stops firing, then the inhibitory neuron stops firing. So then the input will drive the excitatory neuron again. And this process just repeats and repeats and repeats. So the whole system is oscillating. There have been many much more realistic models of this phenomenon, one of which I particularly like from a paper whose senior author was Nancy Capel. a famous neural circuit modeler who is a colleague with my two book co-authors, Emery Brown and Uri Eden. And in that paper, they suggest such oscillatory rhythms accompany states of preparatory attention. This is the kind of interpretation that's pretty widely invoked. Earl Miller's been interested in a related idea that oscillations having frequency roughly around 16 hertz, beta oscillations, could be involved in communication across brain areas during learning and memory, specifically communication between the prefrontal cortex and the hippocampus. The starting point is to look for correlations across trials of the phases of two oscillations in two different parts of the brain. That is, as each phase shifts from one trial to another, we examine the difference in the phases between the two areas. And in Earl's data, it's easy to see noisy but fairly consistent phase differences across the repeated trials. So given a pair of bandpass filters LFP, a standard approach is to compute their phases at a particular point in time in each of the many trials and measure the coupling of the phases, essentially a form of angular or circular correlation across trials. In the last panel on the right, we pick a specific time during each trial at which to measure the phase. So if the oscillatory phases of the two LFPs tend to move forward or backward across trials together, roughly speaking, we observe what's called phase coupling, which would indicate coordinated activity across the two brain areas from which the LFPs are recorded. A commonly used measure is called phase locking value, PLV. The circular diagrams on the right show how it can measure concentration of angles. In this case, the differences in phases across the two filtered LFPs. Under certain restricted conditions, which are commonly found to hold, at least roughly, for neural oscillation phase measurements, there's a strong sense in which PLV is analogous to Pearson correlation for angular, circular random variables. Phase locking value is closely related to coherence, but it's sometimes preferred because it involves only the phase differences, whereas coherence depends also on the amplitudes of the oscillations. In a paper published last year, I think we gave a pretty comprehensive discussion of this and related matters in terms of the multivariate complex value normal distribution. And let me pause and say, this was one of the many papers I've had in my career that improved dramatically between the original submission and the published version. And also, importantly, we leaned heavily on an old paper by Hernando Ambao. There you are. To obtain highly tractable and interpretable results for analyzing shared coherent signals across multiple brain regions using a complex valued version of partial canonical correlation. I wanted to give this shout out to Hernando in part because I remember well his sitting with me and very patiently trying to help me go through one of the basic results in that paper. I don't know if you remember that, but I do. All I can say is, from my perspective, it was enormously helpful, and it's a reminder that sometimes the effort we make to understand things yields its payoff many years later. I've experienced this numerous times in my long career, and this is a great example. So Earl and his postdoc, Scott Brinkat, recorded 24 LFPs across prefrontal cortex, PFC, and 3 regions of the hippocampus during a memory retrieval task. They looked at pairs of LFPs using PLV, but I wanted to develop a multivariate generalization that would allow us to look at all 24 phases simultaneously. And importantly, we wanted a method that would involve a probabilistic graphical model. So just to remind you, a probabilistic graphical model allows us to examine coupling that's unique to a given pair of recordings in the sense that it remains after conditioning on the other recordings. If this little diagram of 3 variables represented a probabilistic graphical model, we'd say that there's a unique, the way I like to interpret it is, there's a unique dependence between x1 and x3, and between x2 and x3, but not between x1 and x2, because the latter's dependence is removed by conditioning on x3. So the problem we set out to solve was how to formulate a multivariate version of phase locking that produces probabilistic graphical models. A great clue for how to do this started from a plot of pairwise data, that is the phase measurements of each of the 2 filtered LFPs. A data histogram is shown at the bottom right. Yellow indicates high concentration of counts. And the bigger plot on the left is based on data that's simulated to be similar to the real data, but it's a little cleaner. It's easier to demonstrate stuff on. If you look at each variable separately, which you can do just, again, mentally projecting down on the x and y axes, these are pretty uniformly distributed, shown here as angles between minus pi and pi, but clearly they are highly correlated. However, there's this weird cluster of points in the upper left corner, which turns out to be easy to explain. In the plot, x1 tends to lead, it's a little ahead of x2. You can see the points are mostly below the main diagonal, which means that when x1 gets to its maximal value of pi, it has to hop back to minus pi. But if x1 is a little past pi, then x2 hasn't gotten to pi. So in rectangular coordinates, the value of x1 in that case is way up the value of x1 is way over to the left, and the value of x2 is up in the top, just below pi. So that's why we see this weird plot. But it's highly unaesthetic, and this kind of thing has always been deeply irritating to me. It's an indication that there has to be a better mathematical formulation. So what do rectangular coordinates represent? They represent points in the product of the real line with itself. The domain of an angle is not the whole real line, it's a circle. And the product of two circles is a torus. And this picture represents one angle in blue, the other in red. So if we make that bivariate plot instead on the torus after smoothing, so yellow again is high concentration, we don't see any aberrant values. One of the students made a nice video to illustrate this, where he placed a red dot to indicate corresponding points in the two representations. So, the torus is really obviously a much more elegant representation. And to get a graphical model for phase coupling analysis, it's statistically natural to develop an exponential family on the multidimensional torus, where we consider the full exponential family having all two-way interactions. In computational neuroscience, this is also called a maximum entropy model. We defined a torus graph to be any subfamily of the full exponential family that has some subset of two-way interactions. And because of that construction, a torus graph is automatically a probabilistic graphical model. It's the analog for random angles of the Gaussian graphical model for random variables in the real line. The rest is a lot of details, which I will spare you, but there's a nice proof of concept which comes from the student's analysis of the phases recorded from the linear probe in hippocampus area CA3. Their torus graph reproduces the spatial neighbor structure of the probe as unique pairwise dependence, whereas PLV, being purely pairwise, cannot produce a probabilistic graphical model, and its graphical diagram could be misinterpreted as indicating unique pairwise independence of every LFP phase and every other LFP phase. Instead, most of those edges are really representing non-unique dependencies. So the full data analytic graphical representations are shown here. Substantively, the main finding is that the apparent dependence of PFC on the dentate gyrus, DG, in the PLV plot can be explained by non-unique dependence through CA3 in the subiculum. This is very intuitive for a memory retrieval task because the dentate gyrus is an area that I think pretty much everybody thinks is associated with input to the hippocampus rather than output. This is a memory retrieval. Okay. So the final example I want to talk about briefly is something we call the Gaussian process current source density estimation. And to set the scene, I'd like to repeat something I said earlier, which is that we ended our review paper by discussing several major issues, and one of them was time-varying dynamics, where we emphasized the statistical assumptions of stationarity. In local field potentials, stationarity is often violated, either because of large transient bursts of activity or because of slow but highly variable processes. Concerning the slowly varying dynamics, we made a comment about what is usually called 1/f noise. This is another topic about which there's been a lot of theorizing in computational neuroscience. We said a conceptual complication, which often produces some confusion, involves the characterization of signals exhibiting long-range dependence as 1/f noise, by which it is meant that the spectral density, the power spectrum, written as a function of frequency f, decreases either exactly or approximately as 1/f, though the terminology often refers to decreases that have a more general power law form, 1/f to the alpha, with alpha not necessarily close to 1. So here's a short excerpt from our explanation. Various realistic considerations, which we talked about, can lead to exact or approximate 1/f to the alpha spectra. However, in practice, a very small number of short-range dependent processes, such as 3 processes, can produce spectra that are statistically and visually indistinguishable from 1/f. We found exactly this behavior in our work on current source density estimation using Gaussian processes. The data analytic purpose of this paper was to get connectivity diagrams involving cortical layers, illustrated here with 10 hertz, that's alpha band phase coupling across 2 different probes in primary auditory cortex. The method's simple conceptually. According to basic quasi-static electrodynamics, the current source density, or CSD, at any point in time and space is the negative Laplacian of the LFP. And the early method for estimating CSD, current source density, was simply to apply numerical second derivatives to the LFP. More recently, it was recognized that CSD estimation is an inverse problem, and that can be solved using the forward model that maps the CSD to the LFP. The forward model is derived from simple biophysical assumptions combined with that Laplacian condition. So we modeled the LFP as the sum of the forward model applied to the CSD plus noise. We then took the CSD to be the sum of 3 components: a deterministic function representing a transient pulse, together with a two-component Gaussian process. As you probably know, standard forms of Gaussian processes describe fluctuations using a single time constant parameter. So here we forced the two time constants, the 2 components, to have very different time constants, making one of them relatively slowly varying and the other more rapidly varying. The first was designed to capture those slow trends, and the second was designed to capture the oscillating fluctuations that we were interested in. So this is what we call the Gaussian process current source density model. The paper provided reasons to believe that this approach is more accurate and more general than other ways to get the CSD. And the alpha band phase coupling that I showed you just a moment ago was not apparent in the raw LFP. So it was an advantage to do this. However, the point I wanted to make now is that the model can reproduce LFP spectral densities quite well, as illustrated in this figure, where the transient part was removed and the remaining 2 components were fit. So this is just one example, and we did not intend to claim that such models will generally fit neural spectra. But in writing the review article, I found an interesting paper by the statistical psychologist Jan Wagenmakers and colleagues, which did make that argument. The fundamental point is this: while there are multiple interesting, I think interesting, intuitive and plausible models of 1 over F spectral behavior, there are many ways to reproduce it, and it's not easy to get strong evidence for one way rather than another. This is a point we statisticians see again and again when reasoning from data is considered carefully. Thus, theorists ought to be cautious when proposing unifying explanations for a particular form of data variation that appears to be present in the data. Okay, that was the main body of my lecture. So I hope that you find what I presented to be consistent with a pair of overarching goals that I want to state. My mission in computational neuroscience, since I first discussed the subject in detail with my eventual close friend and colleague Emery Brown 26 years ago, has been to encourage an appreciation of the teachings of statistics, not just the tools, but their conceptual foundations and the many general lessons we've learned to guide their application. I illustrated the concept of attenuation of correlation and the idea that by introducing a statistical model, we can correct for it by largely removing the noise. Something I didn't mention is that we considered carefully the possible effects of pre-processing steps on inference, and we replicated all the key results in a second mouse. At every opportunity, I try to emphasize the value of using fresh data to replicate well-defined statistical procedures after eliminating steps involving pre-processing and hyperparameter tuning. I also illustrated the fundamental reality that there are often multiple equally good ways of fitting and perhaps interpreting the data by showing how spike count correlation from pairs of neurons could be accounted for, even if the neurons are assumed independent because of population level effects, and by showing how an LFP spectrum could be fitted using a combination of 2 well-behaved processes, thereby reproducing what might be considered 1 over F variation. My mission in statistics has been informed by my deep respect and affection for the aesthetics of mathematics and statistical theory. This is illustrated by our construction of torus graphs, which, by solving an aesthetic difficulty with an elegant statistical framework that we all know really well, exponential families, produced a body of useful methodology. By the way, a really interesting aside about that torus graph paper that we published is that the mathematical part could have been done 30 years earlier. But the statistical and computational implementation relied on recent research, and it couldn't have been done even 5 years earlier, which shows how important the continual progress is to our field. But as I was saying, my mission in statistics has been to understand and evaluate statistical procedures, not only through abstract theoretical research, but also through careful and committed application in scientific settings, where the goal is to figure out how things work. My 3 illustrations all involved PhD-level statistical research using pretty sophisticated math, yet they started with simple intuitions and once understood could be described with simple concepts. The real joy for me comes from this mysterious effectiveness of adapting theoretical ideas to the specific context of real data. Another example goes back to the paper I pulled, from which I pulled a figure at the beginning of the talk. Prior to Giuseppe Vinci's work for his PhD thesis, the literature on spike count correlation had already established that correlation between two neurons increases as the neurons get closer together physically, sort of intuitive, and as their response properties become more similar. So Giuseppe developed a latent graphical model estimation procedure that applied a variation on the ordinary vanilla graphical lasso by allowing a potentially different penalty for each pair of neurons, depending on the values of those two covariates, how far apart they were and how similar their response properties were. So the ROC curves on the right show that the possible improvement here could be dramatic. This illustrates the general notion that it can be very helpful to modify procedures based on existing knowledge. I often say, when it pushes you closer to the truth, bias is your friend. With these final points in mind, I would like to end by recommending to you a breezy 10-page essay you can find on my website. It's an easy read, and people have told me that they enjoyed it because by concisely contrasting statistics and machine learning, I think I was able to present a compact summary of the perspective provided by the field of statistics and what it can contribute to science. So that article was written for statisticians, but I've actually now just recently been expanding it and some related material into a longer, more comprehensive essay, which will form a short book for a general scientific audience. I'm trying to present what I consider to be the most fundamental ideas in our field in words without equations. And I've chosen the title to reflect my understanding of the value and purpose of statistics in science. The title is Reasoning from Data, the Foundation of Trustworthy Scientific Knowledge. My draft currently has 30 chapters, though I prefer to think of it as an extended essay with 30 sections. I'm trying to make it as short as possible. In any case, I mean, each section is like 5 or 6 pages right now, we'll see. In any case, if I had to select a few themes, I would pick these: rigor, context, faith, and progress. We can all appreciate rigor. You can easily guess the kinds of things I'll point to, such as using valid procedures and avoiding p-hacking. Similarly, it should not be surprising that I want to end my essay on the value of statistics for scientific progress, but actually I'm going to have examples throughout the entire essay that show the scientific progress that has come from statistical reasoning from data. But I don't expect you to be able to guess what I have in mind for the middle 2 themes. I feel we don't stress enough the ways in which interpretations of statistical analyses always depend on scientific context. I have become aware, actually relatively recently somehow, I have become aware that failure to recognize this reality is widespread. Many scientists seem to think or act as if statistics steps out of its applied setting to deliver some kind of ethereal rendering of judgment. This seems to me to be at the root of many problematic applications. So I've now started stating one particular aspect of this phenomena as starkly and dramatically as possible. There does not exist a universal context-free method of quantifying strength of evidence. If we could get working scientists to understand this simple lesson from our field, I'm pretty sure it would have a huge impact, and it might also render obsolete the largely, what I consider to be the largely irrelevant diversion of debating about the merits of p-values. My use of faith comes from something I said in a 2011 article called Statistical Inference: The Big Picture, as I was interpreting this figure. Because random variables, and thus statistical models, are abstract mathematical creations, they live in a theoretical world that is fundamentally distinct from the real world of data. We can, of course, do our best to be careful and check the extent to which our theoretical assumptions seem to be reasonable for a particular set of data. But not only can we never be certain, because the assumptions themselves live in an abstract theoretical world, there is no logical way they could be correct. The statement that a model is correct, if taken literally, has no meaning. Thus, crossing the bridge between these two worlds, theoretical and real, might reasonably be called a leap of faith. But it is precisely this leap of faith that makes statistical reasoning possible in the vast majority of applications. Thank you.
Any questions? And also for the people on the line, you can ask the questions. You know, mute yourself and ask questions. A very nice talk. I'm curious about in the first project that you discussed, the correlation and the denoising of the correlation. So I guess, like, intuitively, what drives that denoising? How do I know that it's real denoising?
So if I don't go back to the slide, you know the green dots and the red dots. Yeah. So a simple way to denoise is you just build a model that says the green dots have a correlation. Like let's take a normal, just bivariate normal, okay, for the green dots, and then you say we add noise, another normal distribution. Now you fit a model that has two components. It's just a two-stage, and if it was Bayesian, it'd be a two-stage hierarchical model. You could think of it as a random effects model, whatever. But you know, it's got two pieces: the noise model and the base model for the thing you're interested in. If you fit that to the parameters for the green dots, the base thing, solves the problem.
That's what I guess I'm wondering. The base thing. Right. What is it about in this specific example that it recognizes that this is the base part of the model?
We assume it's bivariate normal. It's just a bivariate normal model. It's got, you know, the usual number of parameters for bivariate normal.
So what if that was just really biological variability? Would you be able to distinguish it versus noise?
I mean, I showed you the naive plot that everybody that I know would do. You can't see anything. I don't know how else you would do it.
Maybe I can ask you.
Yeah, I mean, look, it's a latent variable model, right? I mean, this is the whole thing with latent variables. You can't measure them. So, you know, that's part of the leap of faith, really, is that, you know, you think you're capturing something that's real and it's very intuitive, but you can't actually measure those specific latent variables because they're latent. So you do have to make the assumption that that's, you know, a reasonable representation of the variation. It's an assumption.
Thanks.
Yeah.
Thanks for the very provocative talk. I have my question is, what do you think would be the next methodological challenge we should be looking at? So I'm interested, for example, in can we build models that use concurrently both spike trains and LFPs to decode, to encode, first of all, and secondly, online processing, online estimation, online prediction?
Okay, those are two really different questions, to be fair. So yeah, sure, spike trains, LFPs. I mean, we've done a little of that. Other people have done a little of that. But you know, I mean, my answer to both questions, and I'm afraid it's going to be my answer in the panel discussion too, is, you know, it really is problem specific, right? So So I would first ask myself, okay, when do I want to do LFP and spike trains together? I mean, the case that we did it was when we wanted to relate, when we knew there was an oscillation in the LFP and it was clearly going to be driving those spike trains, but we actually did a whole paper about how hard it is to estimate an oscillation, essentially an unmeasured oscillation from the spike trains alone. We did, I think, a pretty careful study of that. And it's, you know, you can only see it when it's very strong. And most data, it's certainly not visible to the eye. So, so it would be advantageous to actually have the LFP. In fact, the very first paper that Emery did that was decoding, 1998, I think it was, in Journal of Neuroscience, he had the LFP measurements for the hippocampus where there's a strong theta rhythm. You can see it in any LFP, it's just there in the hippocampus. And you have to correct for that because you can't do the decoding of the spike trains unless you take account of this very strong up and down thing. So he had the LFP with, actually I think it was the phase of the LFP that he took with the spike trains. So I mean, that's a simple case where it was done. Now, as far as the decoding goes, again, look, that presumes, you have a purpose for the decoding, and the only one that I know where it's really got to be decoding is brain-computer interface. So for brain-computer interface, anything goes. I mean, I spent, I don't know how many years, probably close to 10 years in that area early on, and then I got out of it because to me, it felt less statistical than engineering. There's a lot of tools you can throw at it. Fine, people just can start throwing tools at it, see what works. It just didn't feel very principled anymore. We did what I felt like were valuable things at the beginning, but so the question back to the, like, okay, when do you want to do decoding? Like a lot, as a lot of machine learning people are very fond of these decoding type things and prediction, and it's like, okay, fine, but for what? Like, I think it's obviously necessary. There's tons of real life places where you have to do prediction. I mean, we can't have our phones do all these magical things if they weren't capable of, if they weren't checked by prediction. But that's not science. Like in science, I don't know where you do prediction. It's not very common, actually. There's sort of, in principle, you're predicting from your scientific theory, but not how most science is actually done, to be honest.
Any other questions?
Okay.
Okay. Thank you, speaker again.